Got it. All righty. Okay, well, I think we're ready to start recording. Thanks everybody for joining us for the March installation of the Symbiota support group. We have a guest speaker today. Before I introduce her and we go into that, we'll just make a few announcements. And of course, if you have any announcements of your own, please feel free to share them with us. I'll pause for a second when we get to the end of our couple slides here. So, first order of business is that, in case you haven't heard, IDIG Bio is hosting its annual digital data conference this year. It will be virtual this time, not in person, and it is scheduled for early June. So, if that's something you might be interested in, just a heads up that that's a few months out, but I'm sure they'll be soliciting for abstracts sooner than later. So, just want to make everybody aware that is happening this year. And second, in case you haven't heard, we will be at Botany this summer, and abstracts are due in well, little over a month, so people have time. And we will be there. A number of us will be there from the Symbiota Sport Hub in early August. Tucson should be great. All thought we would be going there last year, but Palm Springs was awesome, and now we all get to go back to Tucson again, so that's kind of cool. And I guess I said I would pause. Does anyone else have any other announcements before we get into our main presentation for the day? I'm not seeing anything in the chat. Going once, going twice. All right, we shall continue. And also, I should point out, welcome back, Katie. Katie was out last month, but she has returned, and we're very happy to have her back part-time this month. All right, so today's topic is phenological scoring in Symbiota with the USA National Phenology Network, and our presenter is Erin Posthumus, who is joining us from the NPN. She is the Partnerships and Project Manager, and they're based at the University of Arizona. She'll be introducing their Time to Restore project and going over their protocol for phenological scoring using the Torch portal. With that, thank you so much, Erin, for joining us. I'm going to stop sharing my screen and give you a chance to kind of hop on and get set up.
All right, thanks so much. And I am based here in Tucson, so if people are in town for botany this summer, it'll be a great chance to meet you. We are planning to have a booth as well, so yeah, we'll try to get a booth next to the Symbiota booth there.
Sounds great. We would love that.
Okay, cool. All right, so my network, the USA National Phenology Network, as I mentioned, we're based at the University of Arizona. We do a lot of different kinds of work, but most of our work is focused around collecting, storing, and sharing phenology data. We do this primarily through our program called Nature's Notebook, which is a data collection app. But we also work with partners across the country, kind of from regional to national scales, to try to work to inform decisions around phenology, try to advance the science of phenology. And so we have a lot of different kinds of projects. So the one I'm going to be talking about today is our Time to Restore project. This is actually a project that is funded by the South Central Climate Adaptation Science Center. We've actually been funded since 2021 to do this work. And really the inspiration for this project came from partners at the Yuchi Butterfly Farm and Tribal Alliance for Pollinators. We had many conversations with them years ago, and they were really interested in having better information about flowering timing of nectar plants, and then also timing of when seeds are right for collection for restoration purposes. And so we started exploring this idea, and we ended up finding appropriate funding call from the South Central CASC, which is actually based in a regional level covering some additional states. So we contacted some additional partners and kind of centered around this project that would have this regional focus trying to collect information around nectar plants. So the idea we had is, you probably have seen things like this, bloom calendars, planting palettes, that generally will have some information about when flowers are blooming. Sometimes it's at a monthly basis, sometimes it's more of a seasonal basis, like the one on the right, or even just kind of early bloom versus mid or late. And often they're at a state level or regional level. And the information is a little bit coarse. So for your particular location, you might not have a great idea of when things are blooming. So one of our objectives was to try to have more kind of local bloom calendars that would be a little bit more accurate for a particular location. And then the other aspect of this work was to try to understand how flowering timing and seed ripening timing might be changing under future conditions. So this is really our main goal, is to try to provide this information to people working on pollinator restoration, having that local information, and then also trying to understand with increasing temperatures and changes in precipitation, how that might influence the timing of the flowering and the seed. So I mentioned we're kind of working at this regional basis because of the South Central region. So we have our partners in Oklahoma, as well as in New Mexico, the Bosque Ecosystem Monitoring Program. We have partners along the Gulf Coast, the Gulf Coast Phenology Trail, which includes a variety of, is a National Estuarine Research Reserve and National Wildlife Refuges, and several other non-federal partners there. And then we had collaborators at the Great Lakes Indian Fish and Wildlife Commission that we started working with to be able to kind of like help us navigate data sovereignty issues because we wanted to work with tribes and pueblos in this region. And so we have a lot of wonderful partners in the area working towards the same goal. And the two primary ways we're trying to collect data are the program I mentioned, Nature's Notebook, which is our phenological data collection platform. It includes both plants and animals, and it involves collecting information on the same individual plants over time. So you actually will tag a plant or a patch of plants and then go back to those same individuals and collect presence absence data on a number of phenological stages, and then intensity and abundance data as well. But we wanted to supplement Nature's Notebook data with iNaturalist as well. A lot of people really like using iNaturalist. It's really popular in the state of Texas. And so we wanted to be able to have that as an additional way to kind of supplement the Nature's Notebook observations to give us a better geographic spread, but with more incidental kind of observations. And so we focused around 8 different species as our kind of priority species. We wanted to be able to have a kind of a shorter list of species that we would be able to collect data on. We realized that if we collected data on all the species we could that are on the Nature's Notebook list, we'd end up with just a little bit of data on a lot of species. But in order to do the kind of work we want to do with looking into climate drivers, we really had to have a pretty deep data set on a smaller list of species. So we did a number of stakeholder workshops back when we started the project in 2021, and we took into account things like how easy is it to identify the species, how common are the species, are they located across the entire region, and we settled on these 8 species here. And then we actually had started the project in 2021, focused on 3 states, Oklahoma, Louisiana, and New Mexico, but not including Texas, because Texas is so large and we didn't feel like we could really do an appropriate job of covering that state. But then we received some additional funding from the CAST in 2023. So we were able to expand to Texas. And so that expanded our species list up to, I think, 29 species total. So including the 8 that we started with. And just recognizing that Texas had such a great diversity of species, we ended up expanding quite a bit. So this was our list of species that we have kind of as species we're encouraging people to collect data on in Nature's Notebook and in iNaturalist. And we have been also interested in collecting additional data through herbaria as well. So that's where our collaboration with Symbiota comes in. We really wanted to have more of a historical depth of data because we did just start this project in 2021, and to try to look at climate drivers of things like temperature and precipitation influences on plants. It's really useful to be able to have many more years than we have right now, bringing in different years with really different climate and different locations. So we were able to include herbarium specimens as well. And so I think in this example I have here, this specimen is from, I think, 1914. So it's pretty amazing the historical depth that we can add in. And we're going to kind of treat these data similar to the iNaturalist data, where it's basically a single point in time where we know either there's flowering or seeding or not. And that can be another point added to our data set to kind of supplement the Nature's Notebook data. So this activity that we've been doing has actually been undertaken by two students, and we have one of our students who's joined us today, Monica, if you want to give a wave. Monica is here today. So we had Monica Berti-Durso and then Apiram Arshanapalli, who are two undergraduate students who worked on this project from 2024 until last year. They each spent about 300 hours scoring specimens, and we estimate that they scored over 8,000 specimens for 26 of the species in that time. We focused on 6 different collections. We wanted to try to go after collections across the different states. So we tried to have at least one from the different states, although some of them have obviously species or specimens from the different states within the collections as well. But these are the 6 that we scored. And just to kind of go through a little bit of our process, and then I'll jump over to the Torch platform to show kind of how that was done. But basically, trying to get to know these species was the first task. I think Monica and Apiram were not super familiar with a lot of these plants. We don't have many of them here in Tucson. And so they had to kind of first get to know what do these species even look like at the different flowering stages, what do their seeds look like, and how can you tell when a seed is ripe and things like that. So we really leveraged iNaturalist for this. The first task that the students did was to go into iNaturalist and just start looking at the photos of the species to start to get to know the different life cycle stages. And then they put together these really beautiful species rubrics for all of the species that ended up being scored. So this is an example for buttonbush. And this is kind of following the phenophases that we actually use in Nature's Notebook, which is flowers or flower buds to open flowers. And starting from like the very first sign of a flower that you might see on a plant up until as the flowers expand, as they open, where you can see the reproductive structures. We usually put a note here about, how do you know that it's open, with a little bit of detail about that, all the way through to when the flowers are dried. And then we tried to do this for fruits as well. We quickly realized that fruits are very complicated and often really difficult to see, especially if you're looking at a photograph or a specimen, especially if they're like inside of some sort of a capsule. But we tried to put notes on, what the flowers look or what the fruits look like, and then when you might know that they're ripe in a visual way, and tried to make notes about that. We often also would put, something for scale if it was something that was very small, so you kind of have a sense of whether the seed is really tiny versus a little bit larger. And then they also went through just the herbarium, I think the species pages on the Torch portal that we used, just to get a sense of what the herbarium, the digitized herbarium specimens looked like, to see if we might be able to assess both the flowering and the fruiting. And so here you can see some examples where we felt like with buttonbush, it was pretty easy to tell when the flowers were there and even when they were open, because you could see the reproductive structures pretty easily. With the fruits also, you could kind of tell when they were fruits versus flowers because of the lack of those structures. And even sometimes when the fruits were maybe ripe because of them starting to be more crumbly and kind of falling apart. But we also realize it's kind of difficult for some species to tell the flowers and when they're senesced, because sometimes they lose their color, and so it would be a little bit hard to tell whether it was a fresh flower at the time of collection versus one that was already on its way out, kind of dried. And then the seeds as well, we weren't sure if they had lost color or changed color, so that was a little bit more complicated. We also, before we started scoring, did a calibration exercise. This involved, I actually picked out, I think, 18 species where I found an herbarium specimen that had been digitized, brought in the photo into a folder, and then I put together a spreadsheet because we didn't want to be in there in the actual portal scoring until we were ready. So I kind of replicated in a spreadsheet the different drop downs that we had available. And we had 2 different options for scoring phenology. The protocol that was used in the Torch portal was slightly different than the other one that was available from the California Phenology Network. So we did both of them as kind of a test to see what would be kind of easier, what was more complicated, and then also start thinking about which of these would provide sufficient data for our needs. So we did that for all of the species that we had collected as examples. think we had four of us, the two students and then two, actually, I think it was three staff did this calibration exercise. And then we all got together and compared notes and talked about things that were confusing. We ended up deciding that the torch phenology trait protocol had a little bit more depth. It had, something about the relative amount, like mostly buds, mostly open, which we felt like was going to take a lot longer to assess. And we didn't feel like it was going to be providing a lot more useful information than the more simple protocol. So we ended up going with the CCH2 protocol because it seemed like if we were going to be able to get through more specimens, it was a little bit more straightforward. And we felt like it was going to provide sufficient information. So that's the one we ended up using. And I think I have one more slide on this. So just a couple of things that we figured out. We decided to skip answering the questions about the senesced flowers. We were just really unsure of whether we could assess that. If a flower looked like it was kind of dried, we weren't sure if it had kind of happened after collection or if it was dried during the time of collection. So we did not answer those questions. We also skipped a lot of the fruit questions where we felt like it was just impossible to know. So for example, with a Monarda fistulosa, there are these tubes where the seeds sit inside, and we just had no idea in a pressed specimen whether the seeds were in there or not. So we just skipped answering those questions as well. And another thing that was really useful was figuring out the sequence of opening for inflorescences. So knowing if it opened from the bottom to the top or from the inside to the center or outside to the center. It really helped us to be able to assess on an herbarium specimen whether we were looking at kind of buds at the bottom of a structure or fruits that were starting to form. So in the example I have here of the cardinal flower, it's just a little bit easier to know that, oh, these are starting from the bottom and opening towards the top. So you kind of know what to look for in the specimens. So I think, yeah, I'm going to jump over to the portal in a second, but I did just before I close out my slides, I did just want to share that for our announcements, we have a Phenology Week coming up. I can put the link in the chat after I am done sharing my slides. We have a bunch of webinars and doing some social media sharing and things like that, all things Phenology. So we'd love to have you join us for that. And I think this is my last slide. So I would invite you, if you're interested in kind of staying up to date as we start data analysis this summer using the herbarium data, we'll be putting out results in our newsletter. So we welcome you to sign up for that. And just want to acknowledge again our funders for this project, and then the partners that we have and the people that have collected data on the ground as part of Nature's Notebook and INAT. So I'm going to stop my share for a second and jump over to the portal. I don't know if there's any questions so far before we jump over there.
I haven't seen any come through the chat yet.
Okay. Yeah, and Monica, you're welcome to jump in at any time if you want to add anything. I'll do the demo on the portal, but if you want to say anything, definitely welcome you to do that because you did all this work.
Well, it was just like it was a lot of fun working on this project together. I really, really enjoyed like learning about different plants, and then I actually ended up doing like a cross-country road trip, and I got to see these plants in person. So that was like a full circle moment. It was yeah, it was a really great project and definitely one of the best jobs I've ever had. So, yeah, if you have any questions, I'm happy to answer them.
Awesome. Yeah, it was really fun to get Monica's pictures on her trip. She started sending me the Monardas and the sunflowers, and it was really cool. Okay, so we use the Torch portal. We're really grateful to them for allowing us access. So we basically had, at the time we had access to, I think, 6 collections. We've since told one of the collections that we didn't need access anymore, so we don't have that showing here. But our main process was to open up one of our collections. We used the occurrence trait coding tools and then the trait coding from images. And then we would use the CCH2 protocol, as I mentioned. And then we had a process through a spreadsheet that we use for the students to keep track of like who is doing what state and species. So we had a spreadsheet that listed out all the 26 species that we were trying to score, and then the different collections in a column. And then the students would kind of sign up and say like, okay, I'm going to take the Brit collection for this species, and I'm going to focus on Texas first. And so they put like a note about Texas. And so they would put Texas in the drop down here. And then one of the species, so say it was Symbiota, and then load up the images. And the nice thing about this tool was it would just kind of give us all the specimens that were available that had not yet been scored. And so they could see like, when they started, this was like maybe what, 200 or so. Monica, I don't know about for this particular
There
species.
was for this specific species, there was a lot more. And then you would have like, a couple hundred for each state. So we had like the, Texas, Oklahoma, Louisiana, New Mexico, and so you would like, put in each one and then have to go through all of them. It was but it was a lot of fun and it like I think after like the first couple months, it just ended up being like um it was just something really nice to do to like relax at the end of the day.
Kind of meditative. Yeah, so then they would first answer the question about whether it was reproductive, so any sort of reproductive structures, sterile or not sporable. I think pretty often it was reproductive. It seems like for a lot of these species, they were collected with flowers or with fruit. So almost all of them ended up being reproductive. And then they would determine if there were, usually I think open flowers might be a little bit easier than the unopened flowers often, but they would then mark whether the unopened flower was present, absent, open flower. We would skip the senesced and then often skip the fruit, depending on the species. We kind of made determinations about which species we would just totally skip those ones for. And then I haven't done this recently, so I probably would mess this up, but there are a lot of great zooming tools on here as well. So they would zoom in really close into the structures to make the determination and then score. And then when they would save in next, it would bring up the next one. And then once they were done, it was like that was it, and there weren't any more left to score. So it was a really nice system to be able to kind of move through one species at a time for each state, and then mark them off as done. So that I think is what I had planned on sharing. So yeah, are there any questions at this point? We are really excited to use the data. That'll be coming up this summer. So we'll definitely share back at that point any more lessons learned. And then we are planning on also writing up our process here. Monica's put together a nice guide with some screen caps and things. So we'll write up kind of our steps for getting to know the species and the species rubrics and the calibration exercise in case it's useful for anyone else.
I think there were two questions in the chat, by the way, Erin.
Okay, yeah, I see. Are there plans to use this data to track changes in blooming time? Yeah, so that's exactly one of our goals is trying to, I mean, this data in combination with the Nature's Notebook data to try to see if there's relationships between flowering timing and seed timing with certain climate variables. So like temperature, maybe it's, some sort of a seasonal maximum temperature or accumulated precipitation in different seasons. Once we establish relationships between these kind of climate drivers and the phenology events, then we can actually look at climate projections. And so we could say, if there's a relationship where we feel like there's with increasing temperature, flowering shifts a certain number of days, and we can even get it down to like, a certain number of degrees increase, we see a 5-day shift. Then we can actually use climate projections to say, well, if we think it's going to be 3 degrees warmer or 4 degrees warmer in the future in this location, we estimate that the flowering time is going to shift. Sometimes, the onset might shift and the end date might not. So then you end up with a longer flowering period. Sometimes it might be a shortening. So we're not sure yet what we're going to find, but that'll be part of the data exploration this summer. And then another question, is there a protocol available if you want to have our student interns? Yeah. So as I mentioned, yeah, we're definitely going to be writing this up. So I'm happy to share it. I can share it with the Symbiota staff, and you guys are welcome to share it as it's useful.
That would be great. We can circulate it. We could also potentially put it like into the docs or link it like, wherever it fits in best on either on our website or in the docs to make it easier for people to find. So that would be awesome if you're willing to share that.
Oh, yeah, of course.
Do we have any other questions? Thank you so much, Erin. That was a great presentation and a great demo.
So thanks for having us.
We can also, if people have questions that aren't specific to Erin's presentation, we can field some of those now too. Or if people are just feeling like I'd like a little time back, we can kind of end the meeting a bit early. But don't do that quite yet. Just give people a couple more moments.
I have a question for Monica. Monica,
Hi.
what do you think was the hardest part of or the most challenging part of the process, like coming in without knowledge of it
Yeah,
beforehand?
yeah. So I'm actually I'm studying medicine, so I had like no no prior knowledge for it. And I definitely like enjoyed plants and stuff, and I bonded over it and like the medicinal properties. But I think when I first went in, a lot of it was knowing like what structures to look for. Erin definitely trained us really, really well. We had the Nature's Notebook like oh, what was it?
The
Like she gave
photograph?
us like a little booklet and then the
Uh-huh.
yeah, the little modules and stuff to go through. So that was really, really helpful learning what inflorescence was and different different stages. So okay, a flower is not just the ray petals. It's like, oh, okay, we we need to look, this is what an open flower looks like versus like a bud versus a fruit. So that was really, really helpful. And then also I think I because the one on the button bush that Erin showed, I got to make that and I was very proud that she showed that. So I think honestly even just looking through like iNaturalist and looking at all the different stages made me understand a little bit more. Oh, okay, this is probably what different types of this is what a bud looks like and this is all the different variations it could look like. For instance, and so I think that was super helpful too, just like plain old looking at stuff, getting more familiar with it, presenting what I thought things were to Erin, and then her being like, "Yes, this is correct. Oh, this one is not correct. This is actually a bud, or this is actually, a fruit, or this is actually a flower." That was really, really helpful. And then I think the after all that training, I think the hardest like I would say, there's obviously it was a little bit tedious like going through all them, going through, 8,000 specimens that, you know, Abby, he was great. He was rocking through it, but it definitely took a lot of time. But I think that wasn't actually the hardest part. I think it was just like plain old getting trained for it. And then after that, it was a really straightforward thing to do. I could do it while watching TV or something. It was a really great like project to be a part of for sure.
Yeah, I was thinking about we spent so much time doing the rubrics and getting to know the species. I think we probably spent like half of the time doing that. So like probably 4 months of their time and then the scoring after that. But I think it was totally worth it putting all that time into them really getting familiar with what the species look like and yeah, great. And
Yeah, I
because Monica
really.
mentioned, oh, go ahead.
Oh, I was just going to say I really like the point that you have them make some of the training material or like calibration material because that like when you have to teach something, it
Mhm.
suddenly you have to be it really deeply. So that makes a lot of sense to me.
Yeah. Yeah, and I will, Monica mentioned some of the other materials that we have, so I'd love to show those too, just in case they're useful for people, because they're all, freely available on our website. So we have this guide called the Botany Primer. So if anyone wants a PDF guide, this one is, I guess, 79 pages, but it goes through some just basics, and it's all stuff that you have to know for Nature's Notebook. So it still might be useful for you, but we talk about the different, we call them phenophases, life cycle stages. But it's a really nice, like lots of photographs and diagrams and things like that. We end up going through, I can get down to it, different stages. And so we talk a lot about, open flowers, and there's sequences of photos. So this is, yeah, stems and buds. And we go through like what are the different buds with lots of diagrams. We also have an even larger phenophase primer that's really in depth. It's very large, 239 pages. And this one has kind of two sections. One is like really in depth on the different types of stages. And then we go into, for different functional groups, examples of like an entire sequence. So this was kind of the inspiration for the species rubrics. We didn't end up having a lot of our time to restore species in this guide, but I can put the links for each of those in the chat. And then we also have, if you become a Nature's Notebook Observer, create an account, you have access to our observer certification course. And that is what Monica mentioned as well. So if you go down to our module 3, we actually have a lot of lessons that go through, how to identify the different stages, leafing and flowering, and it walks you through with quizzes. So I'll put all those links in the chat.
I just have a curiosity question kind of for the group in general. Have other people used the traits module to score phenological data in their collections, either in CCH2, Signet, Torch, or elsewhere? I know of some collections using it, but it's also not activated in every portal. Not all portals are plant portals, but just kind of wondering, have others used this feature in Symbiota?
I used it several years ago, but I suspect that the protocol has changed as far as where we enter everything, and that's one of the things I was interested in regarding a new protocol.
The tool hasn't changed much since it was first developed in 2019 or so. So it's about the same, but obviously kind of the most important part is developing the protocols specific to the taxa that you're studying.
Thank you.
Chilla asks, are there plans to have something similar for lichens and bryophytes? I think they do use traits in their portal, but it's more for habitat. They use it for different purposes.
Yeah, the traits module is activated in the lichen portal, and there are some traits in there, but they are not phenological. They are mostly, like Lindsay said, about habitat and specific
Like substrate,
kind of,
maybe.
yeah, exactly, substrate specific info. But if those portal communities want to develop any traits that they would want to see across the whole portal, then they're perfectly welcome to. Like if there are, presence of sporophyte versus gametophyte or something, even as simple as that, then that's possible to do.
I think we implemented traits in the bee library as well for wings, maybe. This was a while ago. So there's one insight portal out there that has traits too. But it is not a super widely known tool, I would say, outside of the herbarium community.
I just put a link to the Symbiota docs that talk about the trait scoring from images. And here is kind of the more general article about traits. I don't know if we have, let's see. Traits are a little bit funky to set up in a portal because there aren't any user interfaces where like a super administrator can create traits and then, say whether they're scoreable or not. That's something that you would need to coordinate with whoever is managing your portal. So for most portals, that would be contacting the Symbiota support hub, and we can implement those traits. But again, they would cross the whole portal, so you wouldn't want something that's super specific to your collection. You'd want something pretty generalizable for the whole portal.
Yeah, I was thinking the same thing. That's an important part of setting up that module is you really want to think it through the beginning before you deploy it. It's a little hard to change some of that information on the back end after you start going through it. So it's definitely worth putting the forethought into before deploying it. But once you do, as Erin has demonstrated, it can be quite useful. All right, it looks like we don't have too many more questions. Anybody else for questions or comments? Again, thank you, Erin. Okay, I think we're wrapping up then. Thanks everybody for joining us. We'll circulate the link. I have a feeling this will be a highly rewatched Symbiota support group video. Thanks again. We really appreciate it. And all right, with that, have a great rest of your day, everybody. We'll see you again, same time, same place next month.
Thanks, Erin.
Thank you. Bye.